I'm going to give you a short overview of the pipeline for filarial treatment and give you some update on the ongoing project. So I'm working for DNDi, Drug for Neglected Disease Initiative. This is an organization aiming at developing treatments for neglected tropical diseases and neglected patients. And filarial disease is part of our portfolio, and part of our mission is to bring new treatment. So the filarial diseases are highlighted here. I don't think I need to spend too much time going through these diseases. So current therapies are on this slide. So basically they rely mainly on ivermectin and diethylcarbazamine, which target the microfilari, and they are often given in combination with albendazole. There is a drug targeting adult worm as well. It's Suramin. It's not anymore recommended because of toxicity. And there are new approaches which came lately, which target Wolbachia bacterium, which is a bacterium living inside the worm, and there are some indication that eliminating this bacterium allows to kill the worm or to sterilize the worm. So these therapies have some limitation. So basically all the program directed at controlling these diseases are through mass drug administration. So there is massive campaign to give drug to any patients or to population regardless of if they are sick or not, if they are infected or not. The drugs target the one stage of the parasite, mainly the microfilari, leaving the adult worms alive. And since the adult worm lives for many years, the infection continues. So it requires, therefore, to have a continuous treatment for a long period of time. There are other gaps in current treatment. There are some populations which are co-infected with another worm, Loa, and which produce way more microfilari than Onchocerca volvulus, for instance. And usage of current therapies is not possible or very difficult with those patients because it can trigger adverse events, including encephalopathy. There is also no treatment for case management, so patients who want to access drugs can't. They just have to wait for MDA to come in their region to access drugs. And since the programs have been quite successful, we are advancing through an elimination phase, and even in that phase, there is no alternative to current therapies. And lastly, there is also the risk of resistance, which already occurred in animal health. So the aim of the program here is to develop a selective macrofilaricide, so targeting the adult worm, which can be also implemented in co-endemic region and which could be also available for case management. So here I give a short presentation of what are the main projects from different organizations aiming at developing new therapies for filarial diseases. So if we look at the discovery part, we have the Macro Drug Accelerator, which is led by the Bill and Melinda Gates Foundation, and which encompasses a large consortium of organizations and companies. There are some products which are in preclinical, like oxfendazole and a compound 1066 from, in collaboration from AWOL and ISNTD. There are products in phase 1, Emmadeside and ABV4083. And in the last part of the pipeline, we have some tree therapy which are currently in phase 3, the IDA, called ivermectin, diethylcarbazamine, and albendazole. This is done by a group called DOLF. And very recently, there is Moxie, which has been registered as a new therapy. So what we can see here is that all the projects which are in phase 1 and preclinical have a high risk of attrition, and there is fairly good chance that not any of those molecules will reach the right side of the pipeline. And the other things we can see from this pipeline is that there is a gap in phase 2, phase 3, and towards registration. So there is no many options, actually. So what I'm going to do now is to go a little bit further into these different programs, and I've been break them in two categories. So there are the direct-acting strategy, the direct-acting compound, which target directly the worms, and there are the antibacterial, which I will give you some more indications. So I'm starting with the direct-acting approach. So the first one is moxidectin, which, as I just said, has been registered. It's a project which has been carried out by the Medicine Development for Global Health. And so, as you can see from this graph, it's a recent publication from the Lancet. You can see that moxidectin has a clear advantage over ivermectin, providing way better elimination or reduction of microfilariae. This allows to interrupt the transmission and to relieve the clinical symptom in onchocerciasis. However, despite these very good results, moxidectin has still some limitations. So we have no proof it targets the adult worm yet, so it's still targeting microfilariae. It probably can't be used in lower co-infected patients because it targets microfilariae. And in the same class, it's a macrocyclic lactone, so it's the same class and same mechanism of action as ivermectin. So if there is some resistance occurring, moxidectin can be also at risk. The other product, which is a direct-acting one, is a semi-synthetic product called Emmadeside. It's a product on the market for veterinarian use. It's been originated by Astellas, a Japanese company, and licensed to Bayer. So it's sold as a commercial product in combination with praziquantel or with toltruzril as Procox and Profender. And so DNDi has an agreement with Bayer to develop this compound for onchocerciasis. The challenge of this product is currently in phase 1, and there is attrition in phase 1. We don't know if we will have a therapeutic window and if this product can be moved forward. The other class of product which has been extensively studied in this area is the benzimidazoles, which are very well-known anti-helminthic drugs for gastrointestinal nematodes. So several of these molecules have been studied for filarial indication. Flubendazole has been discontinued for safety reasons. There's been a program from a biotech called Anacor in the US, which is now been acquired by Pfizer, and they come with a boron derivative benzimidazole. You have here the number of their lead compound, 15407. This product has also been discontinued because it didn't have the clear advantage over other benzimidazole. And there is oxfendazole, which is currently in preclinical development by DNDi, but it's a product already in phase 1 by other group for other indications. So we have good hope to start looking for oxfendazole in filarial diseases as well. In the table below, I just put some example of preclinical efficacy of these drugs. So at fairly low drug regimen, you have a complete reduction of parasites, adult parasites. So flubendazole gives sterile cure, oxfendazole almost sterile cure, and IN15407 was a little bit less good. The main challenge of this class of compound, there are tubulin inhibitors, and there are toxicity associated to genotoxicity associated to this mechanism of action, and the challenge is to find a good therapeutic window to move them forward. Moving now to the antibacterial approach, there is this compound I mentioned before. It's a tyrosine analog macrophilaricide. The acronym is Tylamac. So it's an analog of also a well-known veterinarian drug, tylosin. Tylosin is a food additive for dogs, and it's very poorly bioavailable. So AbbVie, which is a large pharmaceutical company, with AWOL, the consortium AWOL, have developed a molecule called AbbVie-4083, and their aim was to increase the bioavailability of the molecule and keeping the antibacterial properties. So basically, as you can see, the modification is not a big modification. There is just this group which has been added in the circle. So what does this molecule do? Basically, it eliminates the Wolbachia bacterium present inside of Onchocerca volvulus or Wolbachia bancrofti, and these inhibit fertility of the worm and possibly kill the adult worm. So this approach is very interesting because we don't target microfilari, and therefore this kind of molecule could be used in co-infected patients with loa-loa. The challenge again here is attrition in phase 1. We need to make sure that we can find a reasonable therapeutic window to move into following clinical development. There are other groups looking for Antiwolbachia product. So this molecule here is being discovered by the University of Bonn in Germany. So it's a natural product issued from I think it's a marine natural product. It's been evaluated against in preclinical models for filariasis and give very good reduction of Wolbachia. So it's currently in preclinical stage. However, since this is a fermentation product, the main difficulties here is to have a process to have large amount of these molecules. So this is a limitation on which the University of Bonn is working. There is another program. This is a collaboration between Eisai and the AWOL group. It's based on small molecule having also an effect against Wolbachia. So in the table here below, I've exemplified few structure from this program. As you can see, there are very potent activity against Wolbachia. And recently they've identified lead candidates, preclinical candidates, which is 1066, which is now in safety enabling studies to move into phase 1. So this is a collaboration between Eisai and AWOL. So again here, I forgot to mention, again, this is a very early stage program, and to go to phase 1, there is still the hurdle of the safety enabling studies, which are also causing a lot of attrition, and therefore we far to have a drug candidate here, just to bear that in mind. So just to come to the conclusion of all of these, there are indeed a lot of research ongoing and some opportunities, but drug development is all about attrition, and there is a lot of failure to get one drug. So to overcome the current limitation, which are current therapies are ineffective against adult worm and require prolonged and repeated treatment, we need to continue looking for new molecules and new drugs. So what we need to have is a macrophilaricidal drug to increase the elimination rate of the disease, or to be able to sterilize the worms for a long duration, for a long period of time, and also to be able to treat patients in co-infected region with Onchocerca volvulus and loa. And for this, I think it's crucial to continue funding the drug discovery research. And on that note, I conclude here.